Welcome to Chapter 3, The Cell. In this section, we're going to look at how scientists discovered the cell as the basic unit of life, how the cell theory was developed, and how microbes were connected to disease. And these discoveries really shaped modern biology and medicine. This chapter covers 4 parts, and we'll try to cover spontaneous generation and start the second one, modern cell theory, in this video. Life ranges from massive redwood trees to tiny microbes, and the microscope first discovered or first used in the 1600s revealed this hidden world and led to cell theory. There are 3 key ideas, and they include 1, that the cell is the basic unit of life, 2, all organisms are made of 1 or more cells, and 3, cells only come from pre-existing cells. Of cell types, we have 2 main ones, and they include the prokaryotes, which do not have a nucleus, and the eukaryotes, which do have a membrane-bound nucleus. And we're going to focus first on the historical discoveries that laid this foundation. This image shows the diversity of microorganisms. Some differ by their size and shape, others differ in their metabolic activity. So they'll look very similar, but their metabolism is different. And all of these differences are important in understanding disease and classification of microbes. In this first section, our goals are to 1, explain the theory of spontaneous generation and why it was once widely accepted, and then 2, describe how different scientists tested, supported, or disproved it. Ancient scholars like Aristotle proposed that life could arise from non-living matter if it contained a vital spirit, something they called pneuma. Examples included things like fish appearing in puddles or frogs showing up after a flood. So this was known as, or still is known as, spontaneous generation, although today we know it's not possible. And back then, they did believe that life could arise from non-life. So through the Middle Ages, people thought that mice could come from moldy grain, or frogs could spontaneously generate from muddy riverbanks. Van Helmont even claimed that mice could form from rags, like imagine dirty cloth, or wheat left in a jar. So this was the theory of spontaneous generation. One of the first, most famous experiments refuting or going against spontaneous generation was completed by Francesco Redi in 1668. So he tested whether maggots appeared spontaneously or came from flies. He put meat in open jars, gauze-covered jars, and sealed jars, and maggots only appeared where flies had access to the meat. So he concluded that maggots must come from fly eggs, not from the meat itself. So this was a really famous early scientific challenge to spontaneous generation. This figure shows Redi's setup. So notice how maggots developed on the exposed meat and even on the gauze, but not in the sealed jars. So the flies can't get in here. So this supported his hypothesis. Next came John Needham in 1745. What he did was he took some vegetable and meat broth and he boiled it briefly, sealed the flasks, but then a few days later he observed microbial growth in the broth, right? They looked cloudy and they probably had a smell. So John Needham argued that microbes arose spontaneously. But the problem is, most likely problem, is that he did not boil the broth enough to kill all the pre-existing microbes. After Needham, Lazzaro Spallanzani repeated the experiments but had better controls. So he boiled broth very thoroughly and found that the sealed flasks remained clear unless he opened them back up and exposed them to air. So he concluded that microbes came from the air, not from spontaneous generation. But still, Needham objected. He argued that boiling so thoroughly destroyed something he called a life force, although nobody really knows what that is. So this debate really kept spontaneous generation alive until around the 1800s. Here we have those 3 scientists. We had Francesco Redi, who refuted spontaneous generation. John Needham, whose experiments supported spontaneous generation because he didn't boil the broth long enough, and Spallanzani, who also disproved spontaneous generation. Okay, our checkpoint. So the theory of spontaneous generation is that life arises from non-living matter if it contains some kind of vital force or a spirit. And this was traced back to Aristotle and also supported by everyday observations back then. Remember, they saw frogs appearing after river floods. They saw mice appearing in moldy grains. They saw fish in new puddles of water. And Van Helmont in the 1600s, remember, he claimed that mice could come from rags or wheat that were left in open jars. And also John Needham, his experiment with boiled broth, where he boiled it briefly, he saw microbes grow, so he argued that they appeared spontaneously. For the second one, challenges to spontaneous generation, we had Francesco Redi. Remember, he had meat in 3 types of jars: open, gauze-covered, and sealed jars, and maggots only appeared when flies had direct access to the meat. Right, so he concluded that maggots came from fly eggs, not from meat spontaneously. And then we had Spallanzani in the 1700s. He boiled broth much more thoroughly in sealed and unsealed flasks, and the sealed flasks stayed clear, no microbes. There's only microbial growth when the broth was exposed to air. So he concluded that microbes came from the air, not through spontaneous generation. But the debate continued because Needham claimed that boiling destroyed some kind of life force that you need for life. By the mid-1800s, the debate still continued, and the Paris Academy of Sciences even offered a prize to resolve this question. Louis Pasteur, now known as the father of microbiology, who happened to be studying fermentation and wine spoilage at the time, was able to show that air introduced microbes, not a life force. And he demonstrated this by first trapping microorganisms in a cotton filter. Louis Pasteur's most famous experiment used swan-necked flasks that he designed, and he boiled broth in these flasks that had these curved necks. Air could come in through the opening, but it trapped microbes in the bends of the swan neck flask. And it turns out that the broth stayed sterile as long as the necks were intact. But if you broke these off, if you opened the flask, then microbes would enter and the broth would become cloudy with microbes. So this really proved that, or showed that contamination came from microorganisms and not from spontaneous generation. Here we can see the dust and other particles like bacteria settling in the bend of the flask, but because of gravity, they cannot get into the broth. But if you break off the neck, within a few days, you'll see that growth occurs. Overall, Pasteur's experiments were thorough and sufficient enough to really provide conclusive evidence that spontaneous generation does not occur. So from that Paris Academy of Sciences, he did end up winning the L. Humbert Prize for these findings. And in some of his lectures, one of the most famous statements that came out from it was that life only comes from life, right? There is no spontaneous generation. Our next checkpoint asks first, how did Pasteur's experimental design allow air but not microbes to enter? So remember, he had those swan-necked flasks. It had a long curved glass tube where air could keep going in and out of the flask, but the airborne microbes were trapped in the bend of the neck and it couldn't go up and into the broth. And this was important because it showed that exposure to air alone did not cause life to appear. So microbe growth only comes from microorganisms, not from that life force. For the second question, what was the control group? So the control group were the flasks where the swan necks were broken off. These flasks became contaminated and the broth turned cloudy because when microbes had direct access to the broth, growth would occur. So this really confirmed that microbial growth is due to contamination from microbes in the environment and not from spontaneous generation. Quick recap. So in section 3.1, we learned how spontaneous generation was once accepted and how Reddy, Needham, Spallanzani, and Pasteur tested it, and ultimately, especially Pasteur, disproved it. So the theory of spontaneous generation was that life can arise from non-living matter if it contains some kind of life force or spirit. And this was proposed by Aristotle and widely accepted throughout the Middle Ages. There were daily observations that really reinforced this, like seeing frogs after floods, finding mice in stored grains, fish appearing in puddles. So back in the day, what they saw, it kind of made sense why they would think spontaneous generation was a reasonable explanation for these things appearing. And even Van Helmont, who was a 17th century Flemish chemist, he claimed that mice could come from rags and wheat in a jar, but he probably mistook these ideal food and shelter conditions for spontaneous generation. So for the certain individuals who tried to really disprove this, it included Reddy, who tested meat in those 3 jars, the open jar, the gauze-covered jar, and the sealed jar, and found that you only had maggots from flies when they had direct contact with the meat. John Needham boiled broth briefly, and he sealed the flasks, but he ended up seeing microbial growth, so he did think that spontaneous generation could occur. But it's likely he just didn't boil the broth long enough, because Spallanzani repeated the experiment, boiling the broth very thoroughly, and he saw that the flasks stayed clear, right? He only had growth if the flasks were exposed to air. And then finally, we had Louis Pasteur with his swan-necked flasks, which allowed air in and out, but trapped microbes in the bend of the neck. And remember, the broth stayed sterile until the necks were broken. So overall, the conclusion is a disapproval of spontaneous generation, and life can only come from previous life. In the next section, 3.2, we're going to turn to the foundations of modern cell theory. So our goals will be to explain the key points of the theory, describe contributions of major scientists, summarize the endosymbiotic theory, and then connect these to germ theory. Modern cell theory has 2 main principles. All cells come from other cells, and cells are the fundamental units of organisms. And Robert Hooke first described cells when looking at cork, but didn't yet recognize their role in life. In the 1830s, Schleiden described plants as made of cells, although he thought they formed by crystallization instead of division. And around the same time, Schwann studied animal tissue and noted the same cellular organization between the 2 types. And together they helped really establish that all living things are composed of cells. In 1852, Remak showed that cells come from the division of other cells. In 1855, Virchow popularized the idea that cells come from cells, right? Cells come from other cells. And this really solidified modern cell theory, but it was controversial because Virchow's work really closely resembled Remak's findings. So Virchow really gained credit for ideas that were actually first presented by Remak, right? And he did not give him any credit. So by today's standards, this would be plagiarism, presenting someone else's ideas without giving them credit. You're basically stealing their ideas and presenting it as your own. And back in the day, at that time, academic integrity standards were pretty unclear, and there was also prejudice against Remak. Today, peer review really helps prevent this kind of misconduct. Here you can see Virchow, who really popularized cell theory, but Remak here, who really provided the original evidence for cell division. Both made important contributions, although credit was not equally shared. Here's our next checkpoint. The key points of cell theory are 1. All living organisms are made of cells. The second, the cell is the fundamental unit of structure and function in living organisms. And 3, all cells arise from pre-existing cells, and this is also known as biogenesis. For the contributions, remember that Remak provided strong evidence that cells come from cell division. And Virchow, he was famous because he had some publications popularizing the phrase that all cells come from cells. And he really made this principle widely accepted, although he didn't give any credit to Remak. And it was really Virchow's reputation and influence at the time that helped spread this idea broadly. Endosymbiotic theory explains how mitochondria and chloroplasts originated, and it proposes that these organelles began as free-living prokaryotes that entered symbiotic relationships with early eukaryotic cells. These relationships are close, long-term interactions between two different organisms, and they can be beneficial relationships, neutral, or even harmful relationships, although the beneficial ones tend to last the longest and evolve over time. Examples of symbiotic relationships include like the gut bacteria, right, in our GI tract. These help us digest certain nutrients, and they also benefit from these nutrients, and they also get a safe place to live. So key evidence for endosymbiosis includes that mitochondria and chloroplasts have traits similar to bacteria. So the evidence is pretty strong. Both mitochondria and chloroplasts have their own DNA and ribosomes, and their DNA is very similar to bacterial DNA. Their ribosomes are like bacterial ribosomes. They have a different type of protein and rRNA makeup that is closer to bacterial ribosomes rather than eukaryotic ribosomes, which are different. These organelles, mitochondria and chloroplasts, also divide by binary fission, just like bacteria, rather than what eukaryotes do, divide through things like the process mitosis or meiosis. And these organelle genes, many of them, have since moved into the host cell nucleus, the eukaryotic nucleus. In 1967, Lynn Margulis revived the endosymbiotic theory with strong evidence from microscopy, genetics, molecular biology, fossils, and even geology. She published a book expanding the idea to explain how symbiosis can drive evolution. And then later, DNA sequencing confirmed similarities between these organelles, the DNA in these organelles, and bacterial DNA. Today, we even observe living examples of endosymbiosis, such as what I mentioned earlier, bacteria in guts, and bacteria is also found in insect guts, or symbiotic organelles in different protists. This figure shows how mitochondria and chloroplasts likely originated from prokaryotes that were engulfed and maintained as partners within the original host cell. So here we can see that in the first endosymbiotic event, this eukaryotic cell is consuming an aerobic bacterium that eventually will evolve into mitochondria. And then over here, we can see that this eukaryote is consuming a photosynthetic bacterium that will eventually evolve into a chloroplast. Here's another checkpoint. So looking at the first question, what does the modern endosymbiotic theory state? Remember that mitochondria and chloroplasts originated as free-living prokaryotes, and these prokaryotes established symbiotic relationships inside these ancestral eukaryotic cells. Over time, they became permanent organelles, essential to modern eukaryotic life. For the second question, what evidence supports the endosymbiotic theory? Mitochondria and chloroplasts have their own DNA and their own ribosomes. The DNA and chromosomes inside these organelles are similar to bacterial DNA. These chloroplasts and mitochondria can divide by binary fission, just like bacteria. They don't go through mitosis. The ribosomes inside mitochondria and chloroplasts resemble bacterial ribosomes instead of eukaryotic ribosomes. And these genes, some of the genes transferred from these organelles to the host nucleus, showing shared integration. And there are also modern examples of endosymbiosis, like the bacteria in the gut and photosynthetic symbionts in protists. That takes us to the end of the first video. In the next video, we'll continue 3.2 with the germ theory of disease.